Welcome everyone to our afternoon session. And we are on our special symposium, The Intersection of Regenerative Medicine and Health Span. And if you've been here over the last 3 days, what you realize is you've been hearing some very unique content. Really the first meeting where we've brought in experts from around the world in the field of the intersection of regenerative medicine, stem cells, and longevity and health span. Because we understand that regenerative medicine is one of the key pillars to moving the needle and bringing health span to the world population. So there are prominent figures in the field, and there are giant figures in the field. And our next speaker, Brian Kennedy, is one of our giants. My name is Bernard Siegel. I'm the Executive Director of the Regenerative Medicine Foundation, and you are now in the 20th edition of the World Stem Cell Summit. We've provided this just recently, recognized the importance of longevity and health span. We just today honored with our National Advocacy Award the Methuselah Foundation. Professor Kennedy has come a long way from Singapore, where he is the professor of the National University of Singapore and director of the National University Health Systems Center for Human Longevity, via way of Germany, to what I like to call the Paris of the Piedmont, Winston-Salem, North Carolina. That's a big schlep, Brian, thank you. In addition, he was the president and served as the president and CEO of the Buck Institute for Aging Research. He has numerous appointments, and of course you can find his biography. This is your studio audience, the World Stem Cell Summit, and of course this will be seen by thousands, and we hope millions, on YouTube soon. Dr. Brian Kennedy.
Thanks a lot. Well, thank you for inviting me. It's great to be here. And you talk about this being the Paris of the Piedmont, right?
Yes.
Well, I grew up in Louisville, Kentucky, so my southern accent is starting to come back already. So
well, I was born in Richmond, Virginia, so I understand that's the business.
It's great to be here, and I'm going to tell you a little bit today about how we're trying to take the knowledge that we've gained in the longevity field and apply it to human models, or human trials, I should say. And so I'm going to take you through some examples of that. There'll be a little bit of mouse studies, a little bit of human studies, a little bit of stem cell studies, and so I'll try to go fast and be clear at the same time. This slide I've been using forever, as several people in the field have, and it really points to I guess the arrow doesn't work. It really points to I think something that most people in the field now have come to accept, that aging is in fact the biggest risk factor for most chronic diseases. And there's a simple next step to that, which is much harder to get the world to accept, which is that if we slow aging or reverse aspects of aging, we'll be able to prevent or delay the onset of these chronic diseases. And to me, it sounds like one implies the other, but it hasn't been so easy to convince people of that. And then we had a little thing called COVID happen, and everybody said that's the biggest challenge of this century, but as you know, COVID mainly caused mortality in people over the age of 65. So it's just another way of looking at the aging problem. Not to mention that aging causes functional decline, and at 56, I can tell you that I can't read the computer screen and see you at the same time, so it's happening to me as well. And so our studies in Singapore are really aimed at health span. And so if you imagine a person's life where they're healthy for a long time, then they get sick, usually what happens is they're not being seen that much by medical professionals while they're healthy. Once they get some chronic disease of aging, then we bring out the armamentarium of approaches to try to treat whatever disease they have. And I think we should treat diseases. But what we're ignoring is the fact that we should be doing health span or health care and keeping people healthy for as long as possible. So our approach is really oriented toward health. And our interventions are done in people that are healthy or close to healthy in middle age, trying to see if we can alter aspects of aging, and I'll tell you how we do that. And so really this story with a small detour into stem cells is going to be about biomarkers and interventions. And I think those are the two things that have really come the farthest in the longevity field over the last decade. We now have hundreds of different kinds of interventions we can think about to slow aging. Very few, if any of them, have been fully validated in humans. Maybe exercise has. But there are many, many, many candidates. And now we have many, many candidate biomarkers to measure aging as well, none of which are fully validated, at least not the modern clocks that you hear of. But many of them are very enticing, and we're using those clocks. And I think our approach can be summarized by saying that the interventions came from one approach, mostly animal models. The clocks came from mostly omic and data analysis. If the two of them interact in the middle and the interventions alter the clocks, I think that does a large part of putting together the different pieces of longevity research and tells us that we're on the right path. So I'll be talking about that. Mostly today I'm going to talk about small molecule interventions, and that's not because I believe ultimately they'll be the best way to go. I think regenerative medicine approaches, gene therapy, many of these things are quite exciting and emerging on the field, reprogramming. But at the moment, most of the interventions, especially the ones we can test in humans, are small molecules or lifestyle. And we're focusing on small molecules. So if you look at the drug age database, there are about 1,000 distinct drugs that have been linked to longevity in some animal somewhere. About 100 of those have been tested in mice. There are dozens of reported effects on health span and lifespan in mice. The ones on the right are things that we work on, and everything in green is something where we've seen a benefit to health span in animal models at least. And so many of these came from other labs, not us originally. And so I think that the good side of this is that there is reproducibility in mouse aging studies. And in fact, we see a pretty good percentage of things that work in our hands when we test them. And we see that kind of as a stepping stone to doing the human clinical studies. If we repeat something and it doesn't work, that doesn't mean it doesn't work. It just means we tested it, it didn't work. But if we can get something to work and it corroborates something that someone else has reported, then I think it gives us more confidence that we have a chance in doing human intervention. So we use the animals for mechanistic aspects of aging, but also as a gateway to doing the human clinical studies. I'm going to tell you a little bit about alpha-ketoglutarate and gemfibrozil today, but you're welcome to ask me afterwards about any of these things. I still think rapamycin is a gold standard in terms of affecting aging, and we're preparing to do a rapamycin study in humans in Singapore in the near future. But I'm not going to talk about rapamycin today. We've also changed how we do animal studies, and I do want to make a comment about animal studies. I think there's, as from the panel discussion we just had, there's a lot of concern as to how well doing things in mice recapitulate what you're going to see in humans. And I think many of those concerns are legitimate, but I want to point out one difference between aging studies and disease studies. Most disease studies in mice are done by engineering a human disease into a mouse. For instance, you create a triple transgenic mouse to study Alzheimer's disease. And then on top of that, they're usually studied in young animals. Alzheimer's is a disease of aging. It's not a disease of youth. And when we try to create diseases in animal models that don't have age-appropriate mice involved, I think we're asking for failure. So one of the things in disease studies that I'm encouraging is that we use age-appropriate mice. I know it's more expensive, I know it takes more time, but think how expensive that failed human trial is compared to the mouse study looking at older mice, and I think it's worth it. The other point I want to make is that by studying normative aging in a mouse, we're comparing natural aging in a mouse to natural aging in a human. There are different outputs for sure, but what the data suggests so far is that the pathways that modulate aging in these two systems, and in fact, many other animal models of aging as well, are at least partially conserved. Things like the TOR pathway, sirtuins, insulin, IGF signaling have been shown to have conserved effects on longevity across a wide range of species. So I think there's a good argument to be made Now, typically what we used to do is study survival in mice, and we're not doing that anymore. We're starting at 18 to 20 months, treating animals for 8 months, and then using frailty and biologic clocks of aging to measure endpoints. One reason for that is it more closely resembles the studies that we're doing in humans, and we want to compare apples to apples as much as possible. The other is that then we can harvest the mice at the end of the interventional study, and we can do a lot more mechanistic studies in tissues, looking at drug action, and a lot of other things you can't do if you just wait till the mice are found dead in a cage. So I think that this is why we've taken this approach. And this is a recent study we did with 4 compounds. I'm just showing you the data from gemfibrozil, but I want to point out a couple things here. There's glycine, gemfibrozil, AKG, and urolithin A. All of them showed benefits in some context, but effects are often sex-specific in mice. And so it's really critical to look at males and females when you're doing aging studies in animals. Second of all, the way we've typically done drug studies in animals is that we pick one dose of a drug, which is done with quasi-scientific methods. And this is not the way to do experiments. So we're not much better. We're doing 2 doses of every drug now. But there are cases, for instance, with glycine, where we see benefits at low dose and detriment at high dose. So I think we need to be more careful about how we're doing these animal studies. If you're doing them short term, you can do more doses too. That's another advantage. So gemfibrozil was beneficial in females. You can see the frailty changes on the right over the 8-month period, both at the low and the high dose. And I want to take you through gemfibrozil because I think it illustrates a couple points that are worth making when it comes to longevity. And one of them is we have to get to drug targets. One of the things we really want to do is to figure out how to combine different interventions together to have synergistic effects. And honestly, having done a lot of that now in an animal, I don't know how to do it. And one strategy was, well, there are hallmarks and pillars of aging. Let's figure out which hallmark or pillar each intervention affects, and then we can just combine them together and we'll get additive effects. And there are people out there in the public that are advocating that strategy. You have people like Brian Johnson that are taking 200 different things a day. And I honestly can't predict what 2 things do together in a mouse. So I want to caution people that I'm on board if you want to be educated and try supplements to slow aging, that's fine. But mixing 3 or 4 things together, I'm just telling you, I have no idea what's going to happen. And they can counteract each other. And so we've taken a different approach that if we know the actual target of the drug, then we might have a better chance of figuring out what works together. And surprisingly, for many, many old drugs, we still don't know what the real targets are. I mean, even with metformin, although a lot of progress has been made recently, we're still arguing about what the targets are. And gemfibrozil is another example. This is a fibrate. It's supposed to activate PPAR-alpha. And I'm going to tell you today that I don't think it activates PPAR-alpha at all. I think that mechanism is wrong. It was published in the 70s and 80s, and I think we have the technology now to really figure out what proteins that drugs are actually binding to. And I'm going to show you that at least part of the effect we see with gemfibrozil is through amino acid uptake. We also have a target for urolithin, and we use this approach called SETSA, which is based on the principle that when a drug binds to a protein, it will stabilize it at higher temperatures. So you can add a drug to an extract, raise the temperature, and do a discovery-based proteomics approach where you find the proteins that are stable at the higher temperature. Doesn't always work, but we've been successful now in 2 cases, and I'm happy to talk more about urolithin A. We think it's another very promising molecule. It works well in our hands. Gemfibrozil we got into originally because it affected yeast lifespan. I don't have time to talk about how we got there. But you can see on the left it extends yeast lifespan and worm lifespan on the right. It also affects health span parameters in worms. And that's what got us excited. We had a conservation between 2 very disparate organisms. This is the data that got everybody else excited about gemfibrozil a long time ago. When you use it in a clinical trial for hypertriglyceridemia, it lowers triglycerides and raises HDL. And this is actually not disputing this. It's an effective drug. It has a few side effects. We're going back and looking much harder into the mechanism. And so this project was started by a postdoc, Chong, all the way back when I was at the Buck, and is now being worked on by Charmaine, who's a graduate student in the lab in Singapore. And so if you look here on the left, can you see? Yeah, okay, so gimfibrazil is supposed to bind to PPAR alpha, free it from an inhibitory complex where it goes into the nucleus and activates transcription of lipid metabolism genes. Even the data that they used to claim this was the mechanism showed only about a 1.5 to 2-fold activation of transcription targets at a very high dose in cells of gimfibrazil. And so we were skeptical about this. And so we did the CETSA, and one of the hits from it was a protein called PEPT1, which is a dipeptide transporter in the gut. We tend to make things simplified, and you might think that you have all these different amino acid transporters in the gut that take in pristine single amino acids for your amino acid requirements in the body. But actually, proteins like this, which is a dipeptide or a tripeptide transporter, and is very nonspecific, actually take up about 70% of the amino acids you get from your gut. And so if you're altering the activity of this transporter, you're affecting amino acid uptake. And so this was identified in 1994. It's on the apical membrane of enterocytes, and it has, like I said, a very non-specific substrates. And we were able to show that when you treat worms with a fluorescent dipeptide, it will go into the intestine, and it goes into these little circles here, which are the intestinal cells. So it's taken up quite well. When you add gimfibrazil, the dipeptide stays in the gut, doesn't get taken up. And the same thing happens when you just knock down this transporter, consistent with it inhibiting this transporter. And also when you add it to human cells in culture, you'll see about a 20 to 50% reduction in most amino acids. And then finally, I think this is the clearest experiment, is when you take out the intestine of a mouse after 2 weeks of treatment and expose it to fluorescent dipeptides, you can reduce uptake of dipeptides into the gut. And then when you look at the serum, this is muscle tissue from the mice, you can see about a 15% reduction in essential amino acids, which are ones that can't be replaced by endogenous systems in the body. And so you don't want a 100% reduction in amino acids to have an effect. A 15% effect is actually a pretty significant change in amino acid levels in the body. So we think this is a mechanism by which gimfibrazil is acting. And if you reduce amino acid uptake in cells, you get reduced mTOR signaling. And a lot of the interventions that affect longevity converge in one way or another on mTOR pathway, and that's what happens here. So if you look at the pink is without gimfibrazil and the blue is with gimfibrazil. In a starved cell, phosphorylation of S6 kinase and mTOR kinase target is dramatically down. When you stimulate with amino acids, it goes up, and gimfibrazil has no effect. If you stimulate with leucine, it goes up, and gimfibrazil has no effect. But if you stimulate with dipeptides, mTOR signaling goes up, and gimfibrazil largely blocks that. So I think we have a pretty compelling story now that we know the mechanism of this drug, which could relate to the hypertriglyceridemia as well, and we're going back and looking at that. We've also identified 2 other targets of the drug that I'm not going to talk about today that are more central to lipid metabolism and also interesting. And in fact, what we're trying to do now is to start a company that derivatizes gimfibrazil, and we want to make an old drug young again by making modified versions of gimfibrazil that better hit the real targets of the drug, because we think they'll be better drugs in the long term. And I've been talking to Owen Phillips, who's the CEO of BrainKey, about this, and we think there's a real opportunity here. People talked a lot about repurposing drugs, and it's been hard to raise money to repurpose drugs because the IP that you get is very hard to defend, and investors get scared by that. But making old drugs new again actually has a lot of appeal because these drugs have been through all the safety studies. We know a lot about them, and if you tweak them, they may do unexpected things, but chances are they're going to behave relatively close to the original drug. And so now we're using AI to go through the drug database, find drugs where we think they have interesting disease indications, may be linked to longevity, and we think the target of the drug is probably wrong. And we're going back and doing this discovery-based approach to try to figure out whether we can identify new targets and how we can make new derivatives that hit those targets better. So if you're interested in that, come talk to me. It is a stem cell conference, so I want to talk a little bit about stem cells, and we have projects on stem cells. And one of the challenges with stem cells is that when you take an iPS cell and you differentiate that into a particular cell type or an organoid, it doesn't behave like an old cell or an old organoid. It behaves like a young cell or a young organoid, making it hard to do aging studies. And so we started this project with Xiamin Zhang at the Buck a long time ago, where we try to differentiate cells, in this case into astrocytes and neurons, and then use in vitro strategies to accelerate aging in those cells so we have a better chance of studying aging. And I'll tell you a little bit about the outcome of one of those projects, just so I'm somewhere in alignment with something happening at the meeting. And so we got interested in astrocytes, and recently the role of cell senescence in the brain has taken off. And one of the things that happens during Alzheimer's disease and with aging is that astrocytes can undergo senescence. And that creates a senescence-associated secretory profile that can affect the function of the nearby neurons, and that may be contributing to how aging affects Alzheimer's disease. So we typically, in our lab, we don't study disease. If we're interested in a disease, we're interested in the aging component. What is it? It's easy to say aging is a risk factor for disease, but why? What is it about aging that makes this disease likely? And is it the same facets of aging that drive all of these diseases, or are they different? These are questions we haven't answered yet. And we may have a clue with this one, so I'm going to tell you about it. And the reason we found this out is we were studying ApoE. And so we started out, I know that you probably know that ApoE4 alleles are a huge risk factor for Alzheimer's disease. You may know that ApoE2 alleles are protective and enriched in centenarians. Most people don't work with ApoE knockouts. I have to say we did it largely because that's the iPS cell we had that was isogenic to our controls to start with. We now have the ApoE2, 3, and 4 isogenic ones we're studying as well. But the interesting thing we found when we knocked out ApoE and did differentiation to astrocytes is that these cells rapidly senesce in our hands. And you can see that with SA beta-gal staining, you can see that with P16 induction, you can see it with P21 induction, and they have a senescence-associated secretory profile and secrete factors like IL-8 and IL-6 and TNF-alpha. So we started asking the question, or Jin-Young Lee did in my lab, what is it about loss of ApoE that might be causing this to occur? And we don't have all the answers, but we have some of them. And surprisingly, it links to nuclear lamins. Now, lamins I've been studying for a long time in my lab in terms of laminopathies. They're in cardiomyopathies and dilated and muscular dystrophies, but they're also associated with progeria, where you have a splicing mutation where you get a dominant version of lamin A called progerin that causes toxicity and causes this really detrimental and rare accelerated aging syndrome where lifespan is reduced to about 13 years. Kids die from heart attacks and strokes, cardiovascular outcomes, and they don't typically have any brain pathology. And the reason for that is there's a microRNA, miR9, which suppresses lamin A expression in the brain. And so when you look in the brain, they don't have this toxic progerin. And it's been shown that if you induce progerin, at least in stem cell models, you can get some toxicity. So this microRNA is protective, at least in the brain. And interestingly, there's two things that happen with lamins, though, with aging. You see an elevated expression of lamin A genes during the aging process. And you can also, so this is two different studies looking at lamin A levels, first in Alzheimer's disease where they're elevated, but also in normal aging. These studies are not very big. I'm relying on some very limited power here, but it suggests that there's alterations where lamin A variants become expressed during the aging process. And so what we found is that ApoE regulates lamin A. So this is a very complicated slide, I apologize, and I'm going to go fast. When you knock out here in the astrocytes, ApoE, you get induction of A-type lamins. You can see that by qPCR looking at RNA. Both progerin and lamin A and lamin C are induced. And when you can also recapitulate the effects by knocking down, I'm sorry, let me say that again. I made this slide too complicated. We know this is through miR9 as well, because if you knock down ApoE, you can induce miR9. And in the ApoE knockouts, you see an induction of miR9. If you inhibit miR9 in a wild-type cell, you can recapitulate this effect in neurons. So what's happening is that ApoE is suppressing expression of miR9 in the brain, and that's leading to activation of A-type lamins. And if you have progeria, also progeria. And so we can take astrocytes from ApoE knockout cells and culture them with wild-type neurons, and we get things like phospho-tau, gamma H2AX in the neurons, and so we can have Alzheimer's-like effects in the neurons by culturing them with the affected astrocytes. We're now looking at organoid cultures to see what's going on. And if you look at longevity interventions like rapamycin, which has been suggested to be protective for Alzheimer's disease, we can actually suppress this effect. So one of the things we see in the absence of ApoE is that you have a loss of synapses, and that's measured by synaptophysin in the brain, and when you add rapamycin, you can bring back that. So that suggests that longevity interventions may be affecting this process. So we're working on the notion that ApoE is important for the aging component of Alzheimer's disease. ApoE levels go down with age. Lamin A comes up because of altered regulation of miR9. That leads to senescence and inflammation in the astrocytes, and that has a knock-on effect on the neurons, which may be enabling them to have more pathology in the context of Alzheimer's disease. I have to say we can recapitulate that, although I'm not showing you today, with ApoE4 astrocytes as well. So it looks like it's consistent with the genetic variants that people see. So there are a lot of questions to be asked here, but hopefully it's starting to tell us what's going on, but the connection between aging and Alzheimer's disease. I want to show some human data, so I'm going to go back now to the clinical intervention studies. And we have the most data on alpha-ketoglutarate, and in the human context, that's a product called Rejuvent, which contains time-released alpha-ketoglutarate and low-dose vitamin A if you're a male, and low-dose vitamin D if you're a female, and that's based on animal data. Originally, we got interested in alpha-ketoglutarate based on worm studies, and then this mouse study where we started interventions at 18 months of age, and we showed that both in males and females, AKG can reduce frailty by about 50% in these animals. Now, it only extends lifespan by 5 to 10%. So what we want to do is argue that this is compressing morbidity, at least in the animals, based on the frailty studies. And that led to the human studies. So I'm not going to show you the mice today, I'm going to show you the human data. And that comes to biomarkers, because the question is, if you're going to do human studies, how do you measure whether you're affecting aging or not? Now, you might say, I'm just going to give this to people and see how long they live. I have yet to convince any graduate students to take on that project. You might do what Nir Barzilai is doing and try to prevent multiple chronic diseases at the same time, and that has the advantage that regulatory bodies can understand outcomes there. It has the disadvantage that it costs a fortune to do that with a drug that costs 4 cents a day. And so I'm supportive of that study. I think we need to do those kinds of studies to measure health span and disease prevention. But I also want to be able to compare and contrast how many different interventions are working in the same context. And I can't spend that much money per intervention. I don't even have enough people in Singapore to do it. So I think that we've turned to trying to co-develop the interventions with the clocks. And so most of you probably have heard about these clocks. You probably heard about DNA methylation clocks, because those are the most developed and most talked about. But I want to argue there's nothing special about methylation. Any omics data that you have with enough people, you can generate a clock that attempts to measure biologic age, either based on starting with the chronologic age of the people, or if you have retrospective data, you can look at the mortality in the population, and both of those kinds of things have been done. So generally what happens is you have chronologic age, and if you're aging well, your biologic age is below your chronologic age, and if you're aging poorly, your biologic age is above your chronologic age. These biologic clocks, they do correspond to each other, but weakly. And so I think that there's a lot we don't know about which clocks are best and what they're actually telling us. I think the best result possible is that each clock is tilted toward different pathways of aging, and so that they're measuring overlapping things, but they're tilted in different ways, and that's why they may not correlate so much. We're still working on that, as are a lot of other people. With the methylation clocks, they generally show the kinds of things you'd like to see. Healthy diets and physical activities decelerate the clocks. Diabetes accelerates the clock. Menopause accelerates the clock. I highly recommend not being a male, because being a male accelerates the clock. Excessive alcohol, which we can discuss moderate alcohol, I have a project on that later, but excessive alcohol is bad. Smoking's bad. Infections, treating cancer with chemotherapeutics, etc. All these things accelerate the clock. So I think it's telling us something meaningful about aging. I think we're still learning exactly what. And so the first thing we did is we had 42 people taking this Rejuvent product for 7 months. And they were willing to do methylation. This is a very basic methylation test using saliva at the beginning of the study and at the end of the study. And the interesting thing that we found is if this line goes up during these 7 months, that means their biologic age was getting younger. And so we saw about a 6 to 7 year reduction in biologic age in this group. Now, is this a great clinical study? No. It's not placebo controlled. And I'm here to tell you that if you're paying $100 a month to be younger, you're probably younger. And so what we're hopefully seeing is that there's some combination of placebo effect and intervention here. This is not definitive, but I think it was hopefully encouraging at least. And there are two parameters that we saw that corresponded to the size of the effect in the individuals that led me to think something real was happening. One is, and this is a little bit surprising, the people that were chronologically older responded more. Now, our original thought was if people get to a later stage of aging, they have multiple diseases, targeting aging may not work as well in that context. But at least with AKG, those people responded better. The other group that responded better were the people that had a biologic age higher than their chronologic age. So if you believe that, the people that were not aging well are the ones that responded better. Now, we don't know if this is going to happen with other interventions. I'm starting to suspect it might with a lot of these interventions. It's not the end of the world. Most people are not aging super fit ways. And so if we have ways of extending health span by a few years and people that are not aging well, that's a revolution in medicine, even with just that effect. So it's good if it's happening, but we're not sure, of course, with other interventions. Now, we went back into the clinically controlled study, and this is an example of the problems with doing clinical interventions right now. It's a bit the Wild West. I don't think anybody knows exactly how to do these studies, and we're learning as we go. Our view is that you have to get in and try and start getting some data to really learn how to optimize. And so we didn't have that last data when we set this study up, so we chose people that were 45 to 65, completely healthy, in Indiana. They exist, we found them, in the middle of a pandemic. So these are also people willing to come into the hospital and do the testing in an infectious disease environment. They're 45 to 65. The interesting thing about them is that this is by Morgan Levine's PhenoAge clock. Now, they were about 6 years younger than their chronologic age at the inception of the trial on average. So we selected an extremely healthy group. And I just told you that's the group that AKG doesn't work as well in, and we found it again. So these are people on 6-month intervention. You're looking at their initial age on the x-axis and the change in aging on the y-axis. And you can see that anybody, this -5 here, most people were 5 years or more younger by this PhenoAge clock at the start, and those people didn't respond to the intervention. If they were within 5 years of their chronologic age, they almost all responded. But it's a very small sample size. And so now we're going back and re-engineering the study design again. This was done by the company. We're doing one now at National University of Singapore with just the sustained release AKG. And we're choosing as an inclusion criteria people that are 45 to 65, don't have multiple diseases, and are biologically at or older than their chronologic age at the start of the study. Is that the right approach? I don't know. But we're getting better as we go, and I think that hopefully we'll learn how to do this. So it's a 6-month intervention, a 3-month follow-up. We have money for about 10 different interventions now. They're much less expensive. This is an RCT study, maybe up to 200 people. Is this going to convince the FDA of anything? No. My point, though, is not to do that. I think we have to generate data that will tell us which interventions are likely to work best in humans, which clocks are going to be linked to them, and how they relate to physiology. So we're not just using clocks, we're using physiologic endpoints, inflammatory endpoints, pulse wave velocity, DEXA, all kinds of other things in these studies. And then we have to start to figure out who's going to respond to what intervention. I don't think there's a single intervention that's going to work for everybody. Even exercise at the same dose is not going to work for everybody. And so we have to understand who's going to respond. And to do that, we need multiple interventions, multiple clocks, and multiple physiologic readouts. And so that's the approach we're taking. Probably that once we find what works best, we may have to go back and do one of these larger studies on health span to get approval. But at the moment, we need data, so that's what we're focusing on. Just quickly, you don't have to just make methylation clocks. People have extrapolated the idea that because you can measure biologic age with methylation, methylation must be the cause of aging. And that's probably not true. I suspect that epigenetic control does regulate aging. I don't think it's the be-all and end-all of aging. And so we made a lipid clock. This was data from worms, just as a proof in principle. But we've gone back now and made lipid clocks from human samples. And they're just as accurate as the DNA methylation clocks. They predict age. This is a control and pancreatic cancer sample. And people with pancreatic cancer are biologically older by this clock than the control population. It's worth noting that it's not just aging that drives disease. If you have diseases that have systemic effects, that's accelerating aging by these clocks as well. So it's a feed-forward mechanism that we're talking about here. And interestingly, if you look at lipid age, you can predict outcome in these pancreatic cancer patients. So the youngest people have a pretty good survival rate in this very debilitating cancer, whereas the oldest people at the study suffer mortality very quickly. So it's telling us something meaningful about biology to make this clock. We've also made a lipid clock from post-mortem brain samples. I think there was a question in the last panel discussion about whether blood's sufficient. I think that what we're trying to do is measure systemic aging, but there's also tissue-specific aging, cellular aging, and all of these things are influencing the aging process. And so one way to look at it, at least I look at it, that a systemic age is like an enabling factor. As long as your systemic age is low and controlled and you're doing well, your body can protect itself against things that might be going wrong. When your systemic age gets high enough, then you're more likely to get chronic diseases, and it depends on what's happening in different organ systems at that time, what you're likely to get. And so measuring organ age together with systemic age we think is going to be valuable. In any case, we made another lipid clock in this case. And the interesting thing is that we were able to make great predictions from about 300 lipids, but then we were able to show that there are only 8 lipids that give you 95% of the predictive value of this clock, and all of those lipids have been linked to aging through other studies in the past. So I think it's one of the advantages of lipids and proteomic clocks is that it's much easier to get the function from these molecules. These are the molecules that have activity in the body. And when you have a CpG island that's methylated, that's also meaningful, but it's harder to get the actual function from that. So what we want to do is make clocks that we can link to pathways and hallmarks of aging so we can understand what the clocks are doing better. And so again, with this clock, we had very few disease samples, but you can see that schizophrenia was associated with an accelerated age. And all of the 5 Downs patients that we had here were dramatically accelerated. So that's consistent with some of the hypotheses out there. Okay, so what are the key questions? I'm going to summarize this. I have no idea what my time is. I love talks where there's no clock for me to look at because I can just talk forever. Huh? 5 minutes? Okay, I can more or less do that. Okay, so I think there are a lot of key questions that we need to answer now. How do we perform human intervention studies? I've commented on that already. I've told you our thoughts. Do we have all the right answers? No. Should we be making these things so that everybody does it the same way right now? I would say no, because we may all do it the wrong way. I think, there's a time to make things systematic. I don't think we're at that time with human intervention trials right now. We need to try our different approaches and see what's working best. Which interventions will work best in which humans? Lots of opinions. Nobody really knows for sure yet. We need to test that. How do we combine interventions? Let me tell you that in mice, there's 3 different outcomes you might get when you combine an intervention together. They might be additive or synergistic. They might do nothing, in which case you see the effect of the single highest intervention, or they might counteract each other. Doing nothing is the most likely outcome. Counteracting each other is the second most likely outcome, and additive and synergistic effects is the third most likely outcome. And I still don't know which ones are going to work until I test them. So there's a lot of work that needs to be done there. Are any of these things going to maximize human lifespan? Enhance maximum lifespan in humans? Or are they just going to square the curve and keep people healthy longer? There are people like Peter Fedichev that would argue, based on the clock data that he looks at, the omics, that we're not going to affect maximum lifespan by these approaches. And that means we have to think about different strategies for how we might affect maximum lifespan. I don't know if that's right or not, but I do think it's important, at least theoretically, to follow this line of thinking, because if it is right, we may be taking the wrong approach to define interventions that have the maximum effect. And so we're starting to think about that. Which biomarkers will be most effective? I've discussed that. And how do we optimize preclinical models to give us the best clinical outcomes? We're trying to do that right now. Again, that's a work in progress, I think. Survival assays have huge benefits in some ways, but they take forever, cost a fortune, and you can't do the mechanistic studies. So we're doing a lot of other things at NUS. We have some deep phenotyping, cross-sectional studies. It's important to note that we have a Malay and Indian population as well as a Chinese population in Singapore, and we know very little about aging in those groups. We've just finished methylation and telomere analysis and associated GWAS and EWAS on 10,000 Singaporeans. And the clocks that we tested 4 different methylation clocks. They tend to work well in the Chinese population. They tend to work very poorly in the Malay population. So we may need to optimize clocks by ethnicity as well. And it's something we're thinking about. There are differences in aging in the different ethnicities as well. And they basically align with socioeconomic status, so that's what you might have predicted. Lots of human intervention studies and other collaborative things that are going on. I didn't talk about reproductive longevity, but I'm also running a center on reproductive longevity and equality that was funded by Nicole Shanahan and the Bia Echo Foundation. And we're interested really in 3 parameters. Why does fertility decline so rapidly, and what's happening with ovaries? And how do we look at postmenopausal outcomes of aging in that context? There's very limited research on ovarian aging, which is a shame because it's a major factor in aging in females, and hopefully we can start to help fill in some of those gaps. And then I think some of you probably know we have a webinar that you're welcome to attend. It's Thursday nights at 7 in Singapore, which means it's 7 in the morning here. I got up and did one this morning. I was very happy to do that this morning. You can join them live if you register at NUS, and you can look my name up in Healthy Longevity, or you can find them all on YouTube after the show. We have 80-some shows now, so you can go find them and look at whatever topic you're interested in. We've had some people in the audience have done the show, so it's, if you don't want to listen to their talk today, you can just go look at the webinar. With that, I just want to thank the people that have been involved. Andrea Myers, my co-director of the Center for Healthy Longevity and really is helping out tremendously on how we do our clinical studies. And we have lots of great collaborators. I'm happy to take questions and thank you.